Welcome back friends. Welcome back to another video session from this channel. So in this video session, we are going to learn regarding genetic code. What is genetic code? What is the genetic code and how the specific code was cracked and who were the aspirants were responsible in elucidating the structure of the genetic code and what are the various salient features of genetic code in detail, right? So what is genetic code actually? How we can define the term known as genetic code? Genetic code, it can be defined as the interaction in between the nucleotide sequence of the mRNA and the amino acid sequence of the protein. Is it clear? So let me explain here. If we consider one particular gene, when it undergoes expression during the transcription mechanism or the transcription process, it synthesizes one particular mRNA, right? So this is nothing but the mRNA. So in case of the mRNA, there is an availability of multiple sense codons which are present very adjacent to each other. So here let us we consider one particular codon known as AUG. AUG is the codon which is encoded by a specific amino acid known as methionine, right? Methionine. So here the specific mechanism or the specific process through which the AUG codon, it is capable to encode or it is capable to recognize the methionine amino acid. It is nothing but what? Genetic code. So genetic code, it can be defined as the interaction in between the nucleotide sequence of the mRNA and the amino acid sequence of the protein. Or in simpler terms, we can define genetic code as the set of rules through which the genetic information which is present in case of a living organism, it may be either DNA or RNA, it gets expressed because of what? Genetic code. The specific mechanism or the specific process through which the genetic information which may be either DNA or RNA, it gets expressed in a living organism because of genetic code. Is it okay? Now, if we consider the specific genetic code, in case of the genetic code, there is an availability of 4 to the power of 3 codons. So let me write it here. How many codons? 4 to the power of 3 codons. That means there is an availability of 64 codons. So out of 64 codons, 61 codons are sense codons. Sense codons. And 3 codons are termination codons. 3 codons are stop codons or termination codons. They are not recognized by any specific amino acid which do exist in nature. So here the specific codon which do exist in nature, it is very specific to one particular amino acid by taking the advantage of a specialized tRNA. So by taking the advantage of a specialized tRNA, the specific codon, it is capable to encode one particular amino acid. Now, how the specific code was cracked? How the specific genetic code it was elucidated here? So prior to that, at first let us we have a look on the various aspirants who were responsible in explaining or in elucidating the structure of the genetic code. So for the very first time, some of the aspirants like Marshall Nirenberg and Hargobind Khorana, right? So let me write it on the board. Marshall Nirenberg and Hargobind Khorana. Marshall Nirenberg and Hargobind Khorana, right? So these were the 2 aspirants who were responsible in elucidating the structure of the genetic code. So once when the specific genetic code, when it was elucidated successfully, then after one more other aspirant known as George Gamo. Who is he? George Gamo. So let me write it here. George Gamo. So he was the person who was responsible for coining the term known as genetic code. Once when the specific genetic code, when it was elucidated successfully, then after George Gamo, he was the person who was responsible for coining the term known as genetic code, right? Now here my question is, how the specific genetic code it was elucidated? What are the various predictions which were made by these aspirants in order to elucidate the structure of the genetic code. So right now we are going to learn the various predictions which were made by these aspirants in elucidating the structure of the genetic code, right? So the very first strategy is nothing but, so what these people they have assumed here. So let me write it here. In the first strategy, in the very first strategy, so these people they have assumed, as we all know, in nature there is an availability of 4 different nitrogenous bases. So in case of DNA there is an availability of ATGC as the nitrogenous base. But whereas in case of RNA, instead of thymine there is an availability of uracil. Is it clear so far? So here let me write it here. What are the 4 nitrogenous bases? A, U, G, C. So these are the 4 different nitrogenous bases which are present in case of RNA, right? So initially what these people they have assumed that here 4 nitrogenous bases they act as one particular codon. Is it okay? 4 nitrogenous bases they act as one particular codon. So in that case, if we consider one particular mRNA, in one particular mRNA, let us we assume there is an availability of 4 codons here. A, U, G, C is one particular codon and G, C, U, A as one more other codon, and the other codon is nothing but U, C, A, G as one particular codon, and there is availability of one more other codon here. So here, as we all know, there is an availability of 4 nitrogenous bases. So here, basing on that, here there is an availability of 4 different sets of nitrogenous bases, right? And these 4 nitrogenous bases they act as one particular codon. Initially, they have assumed that here 4 nitrogenous bases they act as one particular codon. So basing on that, here they have designed 4 different sets of nitrogenous bases. So here when there is an availability of 4 codons, as we all know, one particular codon it encodes one particular amino acid. So when there is an availability of 4 codons, then there is an availability of only 4 amino acids. Then what about the remaining 16 amino acids here? So here the 16 amino acids they remain as useless amino acids. So the reason why this specific strategy, it is not at all applicable. It is a false strategy, right? Then in the second strategy, what these people they have assumed here, what they have predicted here, instead of taking the advantage of 4 nitrogenous bases, they have assumed that here 2 nitrogenous bases they can act as one particular codon. So let me write it here. 4 nitrogenous bases, instead of taking the advantage of 4 nitrogenous bases, 2 nitrogenous bases they can act as one particular codon. In the second strategy they have predicted like this. So let me draw here. So this is nothing but the 4 nitrogenous bases, right? So here there is an availability of U, C, A and G, and here there is an availability of U, C, A and G. So here the specific U as the first nitrogenous base, and it is nothing but the second nitrogenous base. U, C, U, A, U, G, and again C, U, C, 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 A, C, G, right? And again A, U, A, C, A, and again A, G. So let me use some other color here. G, U, G, C, G, A, G, G. So here in the second strategy, instead of taking the advantage of 4 different nitrogenous bases, in the second strategy they have predicted that here 2 nitrogenous bases they are acting as one particular codon. So in that case, under such condition, here they have framed up the particular genetic code. So here there is an availability of 16 codons, right? 4 codons horizontally, 4 codons vertically. So 4 4s are 16. So let me erase the things here. 16 codons. So when they have assumed that here two nitrogenous bases, they are acting as one particular codon, under such condition here there is an availability of 16 codons, right? 16 codons. So here when the number of codons, right, when the number of codons, when it is comparatively lower in comparison to the number of amino acids, so under such condition the specific genetic code, it cannot be carried out or it cannot be worked out, right? Because the number of codons, it should be equal to the number of amino acids at least. But here when they have assumed that here two nitrogenous bases, they are acting as one particular codon, under such condition here there is an availability of only 16 codons. So here the number of codons, it is comparatively lesser in comparison to the number of amino acids. So this specific strategy, it is also not applicable. Then in the third strategy, what these people they have assumed here, instead of taking the advantage of 4 nitrogenous bases and 2 nitrogenous bases, in the third strategy they have predicted that here 3 nitrogenous bases, they can act as one particular codon. Is it clear so far? So when they have assumed that here 3 nitrogenous bases, they can act as one particular codon, under such condition here they have framed up this particular genetic code. So in this particular genetic code, here there is an availability of 4 to the power of 3 codons. That means here there is an availability of 64 codons. So out of 64 codons, 3 codons are stop codons, are the termination codons. They are not at all recognized by any amino acid. So what are the stop codons here? UAA, UAG and UGA. So these are the stop codons, are the termination codons. They are not at all recognized by any amino acid which do exist in nature. So here how the specific code was cracked here? So this is nothing but the first base, right? So it is the first base and it is the second base and it is the third base. Again it is the first base, it is the second base and it is the third base. Again it is the first base, it is the second base and it is the third base. Again it is the first base, it is the second base and it is the third base. So in this way 3 nucleotides, they are acting as one particular codon or they act as one particular codon. So in this way they have designed the specific genetic code. And here in this particular genetic code there is an availability of 64 codons. So out of 64 codons, 3 codons are stop codons. They are responsible only for inhibiting the synthesis of the polypeptide chain during the translation mechanism or the translation process. So the reason why they are named as stop codons are the termination codons. Only 61 codons, they are recognized by 20 amino acids which do exist in nature. Right? So in this way the specific genetic code, it was elucidated. So right now we are going to learn what are the various salient features of the genetic code. Right? So let me erase the things here. How the specific genetic code, it was cracked. So right now we have learned and right now we are going to learn the various salient features of genetic code in detail. What are the various salient features? So let me write it here. Salient features of genetic code, right? So the very first feature, the very first salient feature of genetic code is genetic code is triplet. Genetic code is triplet in nature, right? So what is the meaning of triplet? So as I have already told you, here the specific genetic code, it was framed up by taking the advantage of 3 different nitrogenous bases, right? Here there is an availability of 3 different nitrogenous bases. So when there is an availability of 3 different nitrogenous bases, these nitrogenous bases, these 3 different nitrogenous bases, they act as one particular codon. So the reason why genetic code is triplet. If we consider one particular amino acid known as methionine, right? Methionine. Methionine is the amino acid. Is it okay? Methionine is the amino acid which is encoded by one particular codon known as AUG. AUG. So either in case of prokaryotes or in case of eukaryotes, methionine amino acid, it gets encoded only with the AUG codon rather than any other codon, right? The specific methionine amino acid either in case of prokaryotes. Of course, in case of prokaryotes, there is an availability of modified form of methionine which is nothing but formulated methionine, but still the specific AUG codon, it gets encoded with an amino acid known as methionine either in case of prokaryotes or in case of eukaryotes. And here the specific codon which is encoded by the methionine amino acid is triplet in nature. In the same way, if we consider some more other amino acids like lysine, serine and arginine, right? So lysine, serine and arginine are the amino acids which are capable to encode 6 codons, right? Lysine, serine and arginine are the amino acids which are capable to encode 6 codons in the mRNA. In the same way, isoleucine is the amino acid which is capable to encode with 3 codons. So in all the codons which are capable to encode or which is capable to encode with these amino acids, they are triplet in nature. That means either the amino acid which is capable to encode with one particular codon and the specific amino acid which is capable to encode with more than one particular codon. In all the codons, the codon is triplet in nature. It is not tetraplet or pentaplet. Is it clear? And the next salient feature of the genetic code is universal. What it is? Universal. So let me erase the things here. What it is? Universal. Why genetic code is universal? So let me write it here. So genetic code is universal. It is considered as universal because genetic code, it remains same nearly in all the organisms, right? Right from the unicellular organisms to multicellular organisms and right from the primitive organisms to the complex organisms, in all the organisms, genetic code, it remains same. It remains similar. For suppose, as I have already mentioned you, AUG is the codon which is encoded by methionine amino acid. In case of prokaryotes, the same specific AUG codon, it gets encoded by the methionine amino acid even in case of eukaryotes. The specific genetic code, it won't get varied from organism to organism and from species to species. So the reason why genetic code is universal. Though the specific elucidation of the genetic code, it was carried out by the observations in case of E. coli, but the specific genetic code, it remains universal. It is applicable to all the living organisms which do exist in nature. So the reason why genetic code is universal, right? And the next specialized salient feature of the genetic code is genetic code is non-overlapping. What it is? Non-overlapping. So let me erase the thing here. Genetic code is non-overlapping. What is the meaning of non-overlapping here? Non-overlapping, right? So what is the meaning of non-overlapping? When a particular gene, when it undergoes expression, it synthesizes an mRNA, right? So here there is an availability of one particular mRNA. So in case of the particular mRNA, there is an availability of n number of nitrogenous bases which are present very adjacent to each other. So let us we consider some of the codons like AUG, GUG, GCC or GGC and some other codons like CCG, right? So here why genetic code is non-overlapping? If we consider the very first codon which is nothing but AUG, so here the specific guanosynucleotide, is it clear? The specific guanosynucleotide which is a part of the first codon, it remains associated only with the first codon, right? the specific guanose nucleotide, which is a part of the first codon, it remains in association with the first codon. So this is the first codon and this is the second codon. The specific guanose nucleotide, it won't get associated with the second codon or the next two adjacent nucleotides to form as a codon. Always the specific guanose nucleotide, it remains in association only with the first codon. The specific guanose nucleotide, which is a part of the first codon, it won't get associated with the next two adjacent nucleotides to form another codon. And it is nothing but what? Non-overlapping. So the reason why genetic code is non-overlapping, right? So each and every codon which do exist in case of the mRNA, it is very specific to one particular amino acid. So in this way, the synthesis of the polypeptide chain, it takes place so accurately in case of the various living organisms, either in case of prokaryotes or in case of eukaryotes. So the reason why genetic code is non-overlapping, right? And the next specialized salient feature of the genetic code is genetic code is commaless. What it is? Commaless. So why genetic code is commaless? So as the name itself suggests, if we consider one particular mRNA, here there is an availability of n number of nitrogenous bases, right? So these n number of nitrogenous bases, when they remain in a triplet form, it forms one particular codon. So here, once when the ribosomal subunit, when it gets successfully recruited to one particular mRNA, the ribosomal subunit, it proceeds in 5' to 3' direction. Is it clear? When the ribosomal subunit, when it is capable to proceed in 5' to 3' direction, that means after the successful recognition of the first codon, here there is no availability of any specialized signals in order to recognize the second codon. So after the successful recognition of the second codon, here there is no availability of any specialized stop signals. So without taking the advantage of any specialized signals here, the ribosomal subunit, it is capable to proceed in 5' to 3' direction in order to synthesize the polypeptide chain so accurately, because genetic code is commaless. It do not possess or it do not contain any full stops or commas or punctuation marks. So the reason why genetic code is commaless. Is it clear? Now, so let me erase the things here. And the next specialized salient feature of the genetic code is genetic code is degenerate. What it is? Degenerate. What is the meaning of degenerate actually? What is the meaning of degeneracy? For your better understanding, let me explain it on the board. So in nature, there is an availability of 20 amino acids, right? As we all know, in nature, there is an availability of 20 standard amino acids. So each and every amino acid, it is very specific to one particular codon. Is it clear? So here, according to the degeneracy of the genetic code, if one particular amino acid, if it encodes only one particular codon, right? One amino acid, if it encodes only one codon, under such condition, 20 amino acids, they are capable to encode with 20 codons. Then what about other 41 codons here? So the other 41 codons, they remain as useless codons. So this is not at all applicable. That means in nature, there is an availability of 20 amino acids and one particular amino acid, it is capable to encode with more than one codon. So in such case, 20 amino acids they are capable to recognize with, or they are capable to encode with 61 codons. So as I have already mentioned you, lysine, serine and arginine are the amino acids which are capable to encode 6 codons. Isoleucine is the amino acid which is capable to encode 3 codons. So in this way, 20 amino acids they are capable to encode 61 sense codons. So if genetic code, if it is non-degenerate, right? If genetic code, if instead of degeneracy, if genetic code is non-degenerate, under such condition, 20 amino acids they are capable to encode only with 20 codons. So this is not at all applicable. Genetic code, when it is degenerate, under such condition, one particular amino acid, it is capable to encode with more than one codon. Only under such condition, 20 amino acids, they are capable to encode with 61 codons. Is it clear? So the reason why genetic code is degenerate. And the next specialized salient feature of the genetic code is genetic code is non-ambiguous. Genetic code is non-ambiguous. So what is the meaning of non-ambiguity? Genetic code is non-ambiguous. So what is the meaning of non-ambiguity? So before learning what is the meaning of non-ambiguous, what is the meaning of ambiguous? For suppose, let us we consider one particular codon known as AUG. AUG is the codon which encodes only methionine amino acid, either in case of prokaryotes or in case of eukaryotes. If genetic code is ambiguous, under such condition, the specific AUG codon, though it is capable to recognize methionine amino acid in case of prokaryotes, but whereas in case of eukaryotes, instead of recognizing the methionine amino acid, the specific AUG codon, it encodes with valine amino acid or any other amino acid. If genetic code is ambiguous, but here genetic code is non-ambiguous, that means either in case of prokaryotes or in case of eukaryotes or in case of primitive organisms or in case of complex organisms, the particular codon, it is very specific to one particular amino acid, either in case of primitive organisms or in case of complex organisms. So here under normal condition, the specific strategy it is applicable. AUG codon, it encodes only methionine amino acid, and GCU, alanine is the amino acid which encodes GCU, either in case of prokaryotes or in case of eukaryotes. So the specific strategy it is applicable only under normal conditions, but under abnormal conditions or under in vitro conditions, there are some certain exceptions here. So what are the certain exceptions here? So let me explain you. Under in vitro conditions, if we consider in nature, under natural conditions, the specific UUU codon, it encodes phenylalanine amino acid, right? Phenylalanine amino acid. But under in vitro conditions, that means when the translation mechanism or the translation process, when it is affected by an antibiotic known as streptomycin, right? When the translation mechanism, when it is affected by streptomycin, under such condition, the specific UUU codon, instead of encoding with phenylalanine amino acid, it encodes with isoleucine, leucine and serine. So these are some of the exceptions. In most of the cases, the specific amino acid, it is very specific in encoding one particular codon, but under certain exceptions, in the presence of certain antibiotics or certain drugs, the specific UUU codon, instead of encoding with phenylalanine amino acid, it is capable to encode with some other amino acids like isoleucine, leucine and serine. So it is one of the exceptions here. So the reason why genetic code is non-ambiguous, most commonly in case of organisms, either in case of prokaryotes or in case of eukaryotes, genetic code is non-ambiguous. And the next specialized salient feature of the genetic code is genetic code has polarity. What it is? Polarity. So let me explain here. Genetic code has polarity. So what is the meaning of polarity actually? Genetic code has some polarity. That means it has some direction to proceed, right? When the ribosomal subunit, when it gets successfully recruited to the mRNA, is it okay? When the ribosomal subunit, when it gets successfully recruited to the mRNA, in order to synthesize the polypeptide chain so accurately, the ribosomal subunit, it proceeds in 5' to 3' direction or from left-handed side to the right-handed side. Left-handed side to the right-handed side, right? But never and ever the translation mechanism or the translation process, it gets commenced from the right-handed side to the left-handed side. For suppose if it happens under such condition, what are the various consequences? If the translation mechanism or the translation process, instead of proceeding from left-handed side to the right-handed side, if the translation mechanism, if it is proceeded from right-handed side to the left-handed side, then what are the consequences? If we consider one specific codon known as UGG, right? UGG is the codon which encodes a specific amino acid known as tryptophan. When the specific ribosomal machinery or the translation mechanism or the translation process, when it proceeds in 5' to 3' direction, under such condition, the specific codon is UGG. Is it okay? Under such condition, the ribosomal subunit, it encodes one particular tRNA and it is capable to induce the synthesis of a particular amino acid, which is nothing but tryptophan. But for suppose, if the ribosomal subunit, if it proceeds from 3' to 5' direction or from the right-handed side to the left-handed side, under such condition, the specific codon, it is read as GGU, right? GGU. So GGU is a specific codon which is encoded by a specific amino acid known as glycine. That means genetic code has some polarity or it has some direction. So according to that direction or according to that polarity, the ribosomal machinery, it is capable to induce the synthesis of the polypeptide chain. When the nucleotide sequence of the mRNA, when it is varied, under such condition, the information, the biological information which is present in case of the mRNA, it gets varied. When the biological information which is present in case of the mRNA, when it gets varied, under such condition, the specific amino acid which is very specific to one particular codon, it also gets varied. So the reason why genetic code has some polarity. So all these are the various salient features of genetic code. I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.